All right, good afternoon, ladies and gentlemen. We are going to talk about cell communication today. Cell communication has and signal transduction has an incredibly bad reputation as being the most intimidating little section of our textbook and of AP Biology. The good news is that AP Bio, the College Board, has slimmed down what they expect you to know in terms of details, and they're trying to get you to get the big picture. So I will show you the big picture, and I'll tell you what you do and don't need to know, and I think it will make it very, very user-friendly. So what we're looking at, how cells communicate, what that looks like, blah, 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 blah. So let me go through. I'm going to start I'll get you into the heart of it, and then I'll give you the details. So how cells communicate, it's called signal transduction. So there are three phases, and if all you ever remember from this unit is this picture right here, you probably have learned enough for your AP Bio exam. Of course, I'm probably going to test you on a little bit more detail than that, but not a lot. So let's see. We're going to try to wrap our heads around what this means by going through the rest of the Prezi. So basic idea, cells are capable of signaling each other, and there's numerous ways that they can signal each other, but the signal is called a ligand, Ligand. And the signal comes from outside the cell, and it usually touches a protein that's embedded in the membrane. And you know all about this, right? When proteins have a chemical or a molecule that touches them, it causes the protein to change some kind of shape, which gets transferred. The shape from the outside of the cell to the inside of the cell, that protein that's going all the way through, its shape changes. And when the shape changes, that triggers changes on the inside of the cell. So we have taken a message that was on the inside of the cell, and we've managed to push it through the cell membrane and transfer that message to the inside. Then the message, once it gets into the cell cytoplasm, then it gets transferred through a whole series of reactions and enzymes. It depends. Each one is a little bit different. We call that transduction. So that's when the message is moving from the initial reception of so that's step one, the reception of the message. Transduction means the moving of the message. And then step three is the outcome, the response. Usually the response, I think there's really maybe three that I really want you to know responses. Number one, the response could be something in the DNA where we trigger a new gene, a new protein gets formed because of that. Other cell responses can happen. So other things in the cell can occur because of this signal getting transduced across and triggering something, or apoptosis can happen. So a signal, they even call this signal the death signal. The death signal can touch a receptor and trigger a cascade of events that leads to the death of the cell or apoptosis, and that would be the response. Okay, now you have the overview. Let's go into the ugly details. So how signals are sent. There are local signals and there are long distance signals. The local signals are when the cells are communicating directly with their neighbors. So here's an example on the left. It's called paracrine signaling. You have one cell that has secretory vesicles, so vesicles that are being released, and whatever is inside of them is being released into the environment of the target cells. Then the target cells are receiving those messages and responding to those messages. So that's local signaling. Another local signal is synaptic signaling, and that happens between nerve cells. Nerve cells, interestingly enough, don't actually touch each other. There's a little gap, and you may or may not have studied this in another class. But basically, an electrical signal runs through the first nerve cell, so in this case, coming from the top, and instead of the electrical signal going right to the next one, there's this opening. And so the electrical signal triggers a flood of neurotransmitters to go from the end of the top signal, in this case, the top cell, to across this gap, which is called the synapse, to the next cell. And when the next cell receives those signaling messages, then it starts a whole new electrical signal that shoots to the next one. So that's called synaptic signaling, and that's all local. And here's another example I forgot. So sometimes we learned about this way back in our cell unit. Some cells are actually joined together. Their cytoplasm is shared. And so they can actually send signals between holes in the membrane, and it goes right from one cytoplasm to the next. So the gap junctions in animal cells, the plasmodesmata in plant cells. There's also cell-to-cell recognition, and that's what we're seeing an image of down below, and it's cut off for some reason on my screen. A great example of cell-to-cell recognition is what white blood cells do. So you might remember from our cell unit seeing white blood cells in a video rolling through and kind of high-fiving with lots of cells. So what they're doing in that case, the white blood cell has certain receptors on it, and it's looking to see if its receptors match the proteins that are sticking up, in this case the green things, are sticking up out of the cell if it recognizes them, if they're the same shape. If the cell has a different shape, let's say instead of looking like this, it looks like this. If it has a different shaped protein, then that tells the white blood cell, maybe this cell has been infected, maybe this cell has become cancerous, or maybe this isn't actually a cell and it's actually an invader. Maybe this is a bacteria or a virus. But either way, that signal of the wrong shape tells the white blood cell to attack and to start the immune response. So that's an example of cell-to-cell recognition. All right, those were the close signals, the local signals. These are the long distance signals. And if you're going to remember anything that is an example, I think the endocrine system is the one that I want you to remember. And in particular, I want you to remember stuff about epinephrine. But the endocrine system is basically releasing hormones from various glands in your body. And then those hormones travel through the bloodstream to the target cells. So where they get produced and where they get received may be very, very, very long ways away from each other. You don't need to know any more information than that. All right. So again, you don't need to know tons about this, but I just want to give you some ideas. The ligands, the messengers, the whatever you want to call them, the signal molecules, they're all a little bit different. Some of them are polar, some of them are nonpolar, some of them have this or that. And because you understand this now, I hope you understand this now, when they come in contact with proteins, their shape affects the shape of the protein that they come in contact with. The secondary, tertiary, and quaternary structure of the proteins that they come in contact with changes in response to their chemical makeup. And those changes are what are allowing this signal reception. It's allowing the signal to be received and transferred. In the example here, and you don't need to know much about it, but in the example here, we have actually a receptor that's embedded in the cell membrane, and the signal is received and brought into the cell. In this case, the hormone is lipid soluble, which means that it can actually go through the cell membrane all by itself. It doesn't need a protein there to kind of carry the signal across. So it makes it into the cell, and then it triggers a signal receptor, like maybe in the nucleus or something like that. So that's because it's got different chemistry. So different chemistry is going to impact how the signal is received and who receives it and where it's received. All right, and then try to just look at this. So again, this is just sort of a summary of the different types of responses. If you could remember this diagram, you'd be good to go. So here's our ligand. It's touching our receptor, which is a protein that's embedded in the cell membrane. The receptor is going to change shape, which is going to trigger the movement of this message from one molecule to another molecule to another molecule, sometimes dozens in a row. That's called transduction. So we've moved the message from outside the cell to inside the cell and through the cell. And then that message may even get transferred all the way into the nucleus, where it leads to some sort of a response, like, for example, the formation of some mRNA, which then leads to the formation of a protein. Here's the same exact thing, except this was a hormone. So this one was able to make it in on its own, and then it didn't interact with a receptor until it was inside the cytoplasm. But again, ultimately what it leads to is the formation of a new protein. All right, so let's talk about some of the signaling proteins pathways. They have incredibly intimidating names. So this first one is called GPCR, Ugh. So do you need to know all the details? Not really. But this one is super, 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 super common. You have these in your body, and organisms of all different types have it in their body. The implication, it says it's widely conserved among all domains. Why? The implication is that the GPCRs, that's hard for me to remember the order of the letters, that they evolved super, super long ago in Earth's history, and that those messaging signals are now with all the species that have evolved from those original ancestors that had this message. So basically what you should recognize the letters GPCR. And then let's just take a look roughly at what's going on. Here's a molecule, a protein embedded in a cell membrane. Here is the ligand, the signal molecule. It causes a change in the shape of the receptor and causes another change in another molecule. They bind together, and eventually it triggers, if we get finally to step 4, it triggers a change in another enzyme. And we'll actually look at that new enzyme, this one right here. We'll look at that a little bit later. But that's just an example of one type of reception. Here's another example of a type of reception. And again, you wouldn't yourself need to draw this, but you would need to be able to kind of explain what's going on. You'd need to be able to say that this little red thing is the ligand, and you would need to be able to say that the molecule, the ligand the message has been received, that this is reception that's going on. In these tyrosine kinases, the receptors, so the part that's sticking out of the cell membrane, it accepts these ligands, 2 of them, and when 2 of them get accepted, they bond together in what's called a dimer. The dimer then is activated by ATP, so ATP phosphorylates. Do you see that there's all kinds of phosphoruses that have been added? So we have to have 6 ATP. They phosphorylate that, and then that allows a whole series of cell responses to go from there. So this is, again, a signal reception. We've received the signal, now we've brought it from the outside of the cell to the inside of the cell. And then the last one I think that we'll mention about the reception are the ligand-gated ion channels. Ugh, right? These words are so ugly. But this one really makes sense. Like the name, if you think about it, it makes sense. So ligand, you need to know that word, period. So that's the signal. So ligand gated, a gate is something that opens or closes. Ion channels. So the channels are the proteins that allow molecules to travel through, in this case, ions, because they can't go through the cell membrane because they have a charge. And we know that the tails of the cell membrane of the phospholipid bilayer, those are nonpolar, so the ions can't come through. So in the case of ligand-gated ion channels, when the signal appears, in this case, it could be the opposite, but in this case, it actually opens the gate and allows a flood of ions through, and that leads to some cell response. When the signal, the ligand, goes away, then the gate closes. The whole thing could happen in the reverse. You could have it open under normal circumstances and close when the signal is there. All right, so then we need to talk about second messengers. So the first messenger, that's the ligand itself. But there's some other messengers that are going to carry the message from the initial reception a little bit further into the cell. So cyclic AMP is probably the one you need to know the most. But how much you need to know about it, this right here, too much information. But just listen for a second to see what you need to take out of it. So this is ATP and I gotta think what it is. That is adenosine. There we go. So this enzyme, we always write the name of an enzyme over the top of an arrow that's showing a reaction. So that's just showing you that the enzyme doesn't get used up. It's not part of the reaction. It's just speeding up the reaction. So the enzyme is called adenylyl cyclase. What you need to know is that when adenylyl cyclase is activated, it creates this molecule that's called cyclic AMP. And cyclic AMP is a secondary messenger. Don't worry about any of this stuff here. You don't need to worry about regular AMP. All right, so let's take a look at cyclic AMP and what it might do. So here's our first messenger. Here's the activation. We just saw the same image a moment ago, the GPCR. I'm going to remember Coon Rapids CR because I graduated from Coon Rapids. So GPCR. Now I can say it right. All right, so this is the GPCR right here, and we've activated adenylyl cyclase. That's the enzyme that was above the arrow just now. And adenylyl cyclase, in the presence of ATP, produces this second message that's called cyclic AMP. And that leads to, and now the arrows, you guys, when you see, like, especially when you see more than one arrow, the implication in the diagram is that more than one step is happening there, but they're not bothering to write it because you don't need to know all the steps. So some things are happening that lead to some cell response, but the cyclic AMP is that second messenger. This was the first messenger, the ligand. This was the second messenger. And now we're going to start transducing or transducting, that's not the way to say it, transducing the message through the cell. Another example of a second messenger are calcium ions. How much detail do you need know about the calcium ions? Very, very little. Just look at the diagram for a second. So here's the GPCR. So it's leading to a change in us. Here's a second messenger. And then this second messenger opens up a gate that allows tons of calcium to flow through. And these gates, I'm going to back up for a second, these gates are found in numerous locations within the cell. So you can see them in the cell membrane, you can see them in the mitochondria, you can see them in the endoplasmic reticulum. They're found all over. So these calcium channels end up being super important. So the calcium can push through. That's a second messenger that can lead to all kinds of cell responses, especially when you push a lot of calcium through. Each molecule can lead to another response and can actually amplify what's going on. All right, next, epinephrine. I really want you to know about epinephrine. If we're going to talk about an example of any of this, epinephrine is an awesome example to talk about. So epinephrine is the scientific name. Adrenaline is what you've probably heard of when you hear about somebody getting an adrenaline rush. Many of you think of adrenaline as the hormone that gets released when we're in the fight or flight. So when we're super scared and our heart starts pounding and we're ready to run or we're ready to fight, that's adrenaline. So what's actually happening with adrenaline? So this is an image. I don't think you really need to know this, but it's good to refresh your memory. This is an image of the epinephrine receptor. So this is the thing that's going to accept epinephrine as a molecule. This is the cell membrane. So here are the hydrophobic tails. Notice the alpha helices. I don't know if there's any beta pleated sheets that we can see, but that's the protein that's embedded. And we know that this part of the protein must be nonpolar, or it wouldn't be able to be embedded in the nonpolar tails. And then parts of it are sticking out, and those parts are probably polar. All right, so moving on. Let's take a look at what epinephrine does. So epinephrine is the ligand. It is the first messenger. It goes through the GPCR and leads to this second messenger called cyclic AMP. And then this is what I want you to know, because this is a cool example and it works out really well. Cyclic AMP causes two things to happen, two cellular responses. So I've been telling you, things happen in the cell. Here you totally can understand what I mean by things happening in the cell, and you should be able to say this back to me. So two important cell processes. First of all, it inhibits glycogen synthesis. So what the heck is glycogen? So glycogen is a polysaccharide that mammals, for example, have that they store in their livers. And glycogen is a whole bunch of glucose molecules put together. So the first one of the things it does is it stops storing glucose. So remember, we're having a panic attack. We're either running or about to fight. Something really scary has happened. So we're going to stop storing sugar for a moment, stop storing glucose, and it promotes the breakdown of glycogen. So glycogen has all these glucose molecules. We're going to suddenly start pulling those glucose molecules off of the glycogen. Well, what does that do? That floods our cells, and then it leaks out. It floods our cells with glucose. And so glucose is going to be absorbed by it's going to be broken down, and then the mitochondria is going to do the cell respiration, and we're going to produce a ton of ATP that allows our muscles to either, book it out of there or to fight for our lives. So that's what epinephrine is doing to us. Now, here is something that I think is interesting, still part of this epinephrine idea. Epinephrine is the ligand, But there's a whole bunch of different effects depending on where the epinephrine is received or what kind of receptor it gets. So let's take a look at the diagram. You need to understand the diagram. It's not complicated. So here, look at the receptor. It's called a beta receptor, and it's green in the diagram. Over in this diagram, there's an alpha receptor, and it's blue. Notice that the beta receptor here is on a liver cell, and here it's on actually a blood vessel. It's the same receptor, but it's on two totally different places. So we're going to see how the response is different, even though it's the same ligand and it's the same receptor, because they're located in different places, it's going to produce a different effect. And then over here, it's the same ligand, but a different receptor on the same thing. It's both blood vessels. And we're going to have yet another response. So let's walk you through it. So here's epinephrine. It touches the receptor, and it leads to the breakdown of glycogen so that we produce a flood of glucose. So now our body can make lots of ATP through cellular respiration. So we've got lots of energy. That's one effect. Here's the same ligand, the same epinephrine. Now it's going to touch the same type of receptor that's found on the outside of blood vessels. And when it touches the blood vessels, it's going to cause the blood vessels to dilate. If you look, that means the opening on the inside gets bigger. Why would it want to do that? Because that allows the flood of more blood to go through them, which brings with it more oxygen and more nutrients so that they can get to your cells faster. So your cells can do more cell respiration. So this is all about you being able to run really fast and get energy and get oxygen to your cells as fast as you need. The third response, here's epinephrine again. This time it's a different receptor, but still on blood vessels. Oh, and what I forgot to say, because this is the important difference, in this case, the blood vessels are your skeletal blood vessels. So these are the skeletal and muscle. These are the ones that are going to help you with fighting and your large muscle movement. The blood flow has increased to them. In this case, epinephrine actually causes the blood vessel to constrict, to get smaller. Well, when would we ever want that when we're panicking? We want the blood flow to decrease to our digestive system. Right now, that's not our priority. Digesting food when we're in an emergency situation, not important. So your blood is actually being shunted to the parts of your body that need to get you out of there or allow you to fight. And less blood is going to be flowing to your intestinal system. So those are the three different effects. Those are three different effects of epinephrine. And I think you should be able to say that or understand that as an example of signal transduction. All right, so not only can we receive a signal and transfer it into the cell and carry it through the cell and lead to a response, we can even amplify the response. So let's say you just have one signal. I'll show you the example. I'm mumbling right now. So here's our one signal molecule. It touches a receptor. Now, you do not need to know the details on this. And this is where students get bogged down and horrified. I'm just going to tell you, it's not worth your time. You might see a question on it, and it's not worth your time to spend learning about it. So basically, this is called a phosphorylation cascade. So you can see that ATP is getting used, and we're adding a phosphate. Here's the phosphate that gets added. ATP gets used, and we add another phosphate. ATP gets used, and we add another phosphate. That's why it's called phosphorylation. It's a cascade because it's one molecule affecting the next molecule, affecting the next molecule, affecting the next molecule. That is actually increasing, and I'm going to show you an example, increasing the message. We just had one signal molecule, and now we're going to have a bajillion messages on the inside, and I will show you how that works. So take a look at the numbers here. We start with one molecule. As we start going through the phosphorylation cascade, we have 10 to the 2 molecules, then we have 10 to the 4 molecules, then we have 10 to the 5 molecules, then we have 10 to the 6 molecules. We just went from 1 molecule to 10 to the 6 molecules. This signal exploded inside of the cell and allows your body to respond incredibly fast, or allows your cell to respond incredibly fast. And so you have this enormous, in this case, the response, the glycogen into glucose 1-phosphate, 10 to the 8th molecules in a split second because of this phosphorylation cascade. That is all you need to know about it. Don't worry anymore about it. And that's a form of positive feedback. As one molecule is made, it's received by another and tells the next step to increase, and then the next one increases, and it keeps increasing. So if the products are increasing, we call that positive feedback. It's activating it. That's positive feedback. All right, it can get more complicated. How much more information you need to know here? Not positive. Let me just walk you through it really quick. So notice in these 4 diagrams that you have the same signaling molecule each time, but what happens with that signaling molecule is different. And we just kind of saw that in epinephrine. And so if you can remember that with epinephrine, I think you're good. So in this example, the receptor leads to one particular type of cell response. In this case, the receptor activates a different protein or enzyme that splits, and now we have 2 pathways. They call it branching, 2 pathways and 2 cell responses, not unlike the glycogen that gets stopped, that gets inhibited for building glycogen, and that activates breaking down glycogen. That's a branching in that case. Here, we just learned about this in the last unit. This molecule can lead to, so this ligand can lead to activation or inhibition. So this would be like an allosteric inhibitor or an allosteric activator. This is the molecule that either permanently turns an enzyme on or permanently shuts an enzyme off. And so that leads to different responses depending on whether it's been activated or inhibited. And then my picture is in the way. Let's see if I can move it. Maybe my finger. There we go. So then in this last one, we have a different receptor this time. So notice that the receptor was the same each time. This time we have a different receptor, and that's leading to a different cell response. And that's not different than what we just talked about with the keep going. What we just talked about with epinephrine. Okay, we are almost done, peeps. Here's the good news. This Prezi goes into a lot more detail, but as I study over the AP College Board requirements, the only one of the examples that they mention is apoptosis or apoptosis. So I think you should know epinephrine as an example, just to have one in your back pocket that is easy to understand, that you can discuss if you need to. And then you definitely need to know apoptosis. Quorum sensing and yeast mating, they're interesting, but you don't need to know too much about them. So I'm just going to zip through them. We're not even going to talk about them. Oh, I could have done this more efficiently, couldn't I? Oh, well. Let me get to apoptosis. Okay, so remember that apoptosis is programmed cell death. It's programmed because the signaling pathway that it is because of the signaling pathway, it's already there. It's already present. So I think at one point it says, let me show you this here really quickly. It says, notice that death proteins, they seriously call them death proteins, are present in an inactive form prior to signal reception. Why are those death proteins there prior to signal reception? So that it can happen quickly, so that it can happen, so that apoptosis can take place quickly. So here's a death signal, a ligand. It activates a whole series of events that happen inside of the cell. Do you need to know them? No. And you may or may not remember this from when we talked about apoptosis before, but it leads to sort of the cell losing its shape, and it produces like these bubbles, and they call them blebs. And yes, that's the official term, bleb. So that death signal leads to apoptosis. It causes that cell to die. Ladies and gentlemen, that is it. Let's go just take a look at these questions. So these are always good questions for you to review, but I never want to take time because I feel like I've already done so much on the video. So take a moment to see if you understand the questions and let me know if you have any questions. That's it.